Deep in the tropical rainforests of Thailand, Brazil and Central Africa, a war is being fought that most humans will never see. It is not a war between predators and prey in any traditional sense. It is a war between a fungus and an ant, and in this war, the fungus does not simply kill, it takes over. It invades the ant's body, hijacks its muscles, overrides its decision-making, and forces the insect to carry out a series of precise and deliberate actions that serve only the reproduction of the fungus itself. The ant becomes a puppet, a vehicle, a dead organism walking through the forest on legs it no longer controls. This is the story of Ophiocordyceps unilateralis, one of the most sophisticated and genuinely terrifying parasites ever documented in the natural world. To understand how this parasite operates, we must first understand its host. Carpenter ants of the genus Camponotus are among the most common and most socially organized insects in tropical forests around the world. They live in large structured colonies, sometimes numbering in the tens of thousands, typically located high in the forest canopy, where conditions are warm, dry, and relatively safe from ground-level threats. Each colony functions as a single coordinated superorganism, with workers assigned to specific roles, including foraging, nest maintenance, brood care, and defense. Communication between colony members is maintained through a complex system of chemical signals called pheromones. Every ant knows where to go, what to do, and when to do it. The colony operates with extraordinary efficiency and discipline, and it is precisely this order that the fungus exploits. The infection begins when a carpenter ant worker, traveling along the forest floor during a routine foraging trip, unknowingly steps on or brushes against a microscopic fungal spore released by a previously infected ant that died nearby. The spore is impossibly small, often less than 1/100th of a millimeter in diameter. It attaches to the outer surface of the ant's exoskeleton and immediately begins to germinate. Using specialized enzymes, the spore dissolves a tiny hole through the hard chitinous exterior of the ant and enters the body cavity. Once inside, the fungal cell begins to multiply, slowly at first, and then with increasing speed. For the first several days after infection, the ant shows no visible signs of illness or behavioral change. It continues to forage, communicate with colony members, and return to the nest as normal. From the outside, it appears to be a perfectly healthy, fully functioning member of the colony. But inside its body, the fungus is growing rapidly. Fungal cells called hyphal bodies are proliferating throughout the ant's hemolymph, which is the insect equivalent of blood. These cells are spreading to every organ, every tissue, and every muscle group in the ant's body. The fungus is mapping the ant from the inside, learning the architecture of its host before it makes its move. What happens next is where the story becomes truly disturbing. At a specific point in the infection, typically after 1 to 2 weeks of silent internal growth, the fungus initiates a dramatic behavioral takeover. The ant, which has been behaving normally up to this point, suddenly begins to act erratically. It leaves its regular foraging trail. It wanders away from the colony. It staggers and stumbles as if confused or disoriented. Most critically, the infected ant begins to do something that healthy carpenter ants almost never do. It climbs down from the canopy toward the forest floor. This is significant because the forest floor is not where carpenter ants are supposed to be. Their colonies are in the canopy, their food is in the canopy, their entire social world is in the canopy, and yet the infected ant descends, moving against every instinct and behavioral pattern that evolution has programmed into its species over millions of years. The fungus is driving, the ant is merely the vehicle. Researchers have discovered through careful study that the fungus does not actually infect or directly control the ant's brain. Instead, it surrounds the brain with a dense network of fungal cells that influence its chemical environment. The fungus secretes specific compounds that alter the concentrations of neurotransmitters in the ant's nervous system, effectively changing the chemical instructions that the brain sends to the muscles. The ant's brain remains technically alive and intact, but it is swimming in a bath of fungal chemicals that override its natural impulses and replace them with new commands that serve the parasite. The precision of this manipulation is extraordinary and deeply unsettling. The fungus does not simply make the ant wander randomly, it guides the ant to a very specific type of location. Research conducted in tropical forests has shown that infected ants consistently seek out locations that share a narrow set of environmental conditions. The ant will find a leaf or a twig on the north side of a plant, approximately 20 to 30 centimeters above the forest floor, in a zone where temperature stays between 20 and 30 degrees Celsius and humidity remains above 94%. These are the exact conditions that the fungus requires for its reproductive structures to develop successfully after the ant dies. The ant is not choosing this location, the fungus is choosing it through the ant. Once the ant reaches the optimal position, the final and most iconic stage of the infection occurs. The fungus compels the ant to perform what scientists call the death grip. The ant clamps its mandibles down onto the underside of a leaf vein or a twig with a force so powerful that it locks permanently in place. Microscopic examination of the jaw muscles in death grip ants has revealed that the fungal cells have physically invaded and destroyed the muscle fibers in the mandibles, causing them to contract and lock irreversibly. The ant could not release its grip even if its brain was still sending normal signals to its jaws. The muscles have been structurally hijacked at the cellular level. The grip will hold the ant's body in position long after it has died. Within hours of the death grip, the ant dies. The fungus has consumed nearly all of the soft internal tissue, leaving only the hollow exoskeleton attached firmly to the leaf or twig. Now the reproductive phase begins. Over the following days, a long thin stalk called a stroma emerges from the back of the ant's head, growing upward and outward from the dead body. This stalk eventually develops a capsule at its tip that is filled with thousands of new fungal spores. When the capsule matures, it bursts open and releases the spores into the air. The spores drift downward to the forest floor below, scattering across the ground where other foraging ants are walking. The cycle is ready to begin again. The entire process, from initial infection to spore release, takes approximately 2 to 3 weeks, and it has been happening in tropical forests for an almost incomprehensible span of time. Fossil evidence of the death grip, preserved as distinctive bite marks on ancient fossilized leaves, has been dated to approximately 48 million years ago. This means that Ophiocordyceps has been manipulating ant behavior for at least 48 million years. The parasite was perfecting its mind control strategy while the earliest ancestors of modern primates were only just beginning to appear on Earth. Carpenter ant colonies have not been passive victims throughout this long evolutionary arms race. Over millions of years, they have developed behavioral defenses against the fungus. Worker ants are known to identify infected colony members and physically carry them far away from the nest before the fungus can complete its behavioral manipulation. Some ant species have evolved the ability to detect fungal spores on the bodies of returning foragers and groom them off before infection can establish. Entire colonies have been observed relocating their foraging trails to avoid zones on the forest floor where high concentrations of fungal spores are present. The ants are fighting back. But the fungus continues to evolve alongside them, refining its methods of infection and control in an endless biological arms race with no final winner. Perhaps the most unsettling aspect of Ophiocordyceps is what it reveals about the nature of individual identity and autonomy in the natural world. We tend to think of an organism as a single unified entity that makes its own decisions and controls its own actions. The zombie ant challenges this assumption in the most fundamental way possible. Here is an animal that walks, climbs, navigates and performs complex physical tasks, all while being piloted by another organism entirely. The body belongs to the ant, the behavior belongs to the fungus. The question of who the ant actually is during its final days becomes genuinely difficult to answer. There are over 400 known species of Ophiocordyceps fungi worldwide, each one specialized to infect and manipulate a specific host insect species. Some target beetles, some target moths, some target other ant species. Each fungal species has evolved its own unique set of chemical tools and behavioral manipulation strategies tailored to its particular host. The zombie ant fungus is not a single isolated oddity. It is one member of a vast global family of mind-controlling parasites, each one independently perfecting the art of hijacking another organism's body for its own reproductive benefit. Deep in the forest right now, an ant is leaving its trail. It does not know why. It walked this path 1,000 times before and never once deviated. But today something is different. Its legs are carrying it in a direction it has never chosen to go. Down the trunk, away from the canopy, away from the colony that raised it and the trails that defined its existence. It moves toward a leaf it has no reason to visit. A leaf on the north side of a plant, 25 centimeters above the damp forest floor, in a zone of perfect humidity and perfect temperature. Perfect not for the ant, but for the fungus that has been quietly rewriting its behavior for days. Its mandibles are tightening around a vein. It has no reason to bite. The grip is final. Inside its body, something ancient and patient has been guiding every step with chemical precision, perfected over 48 million years. The ant will die here, clamped to this leaf in the humid shade of the understory. And in the days that follow, a thin stalk will rise from the back of its head, reaching upward like a finger pointing toward the canopy. At its tip, a capsule will open, and from it, new spores will fall like invisible rain onto the forest floor below, drifting silently downward, waiting for the next footstep.